Okay. Uh, Good morning, people. So last class we were looking at the structure of the kidney, and of course we were looking at the longitudinal section of the kidneys as well. Like in terms of location and everything, you are very well aware now, I have suppose, where the kidneys are located. Also, in terms of the section we were discussing about, when you cut it vertically, what you get is these conical shape structures which we call as medulla, or this whole region is called as medulla, which is internal, and the outermost region is called as cortex. And like it was said, the cortex is actually more darker than the medulla, unlike it is shown in this diagram. And the reason being because the cortex has most of the nephrons, which has like very well connected blood vessels, and that's why it will be more darker compared to the medulla portion. So that was renal medulla and renal cortex. Uh, then what else we had looked at was yeah, this presence of the sphincters around the urethra, like at the base of the urethra, or like at the bladder into urethra, or even at the ureters, which are the opening from the uh, kidneys to the urinary bladder. So that's where the sphincters were present, which will control the process of urination, what is scientifically known as micturition. Now, and of course, the single units which are present, which are tubular ones present within the kidneys, they are called as uriniferous tubules or nephrons or renal tubules and so on. Right? Now, we will continue from there. So nephrons, as you know, these are the structural and the functional unit of the kidney. That means they make the structure of the kidney. That's why the structural unit, and the functional unit because they also perform the function of the whole of the kidney. That's why they are called as structural and functional unit of the kidney. Now, the uh, basic unit, that is the nephron, it consists of glomerulus. So, where is that diagram? So, there is something. So, this is how the structure of nephron would look like. So, nephron, you can imagine there are millions of these within the kidney. So, when we say that this is the kidney structure, or this is the internal structure or longitudinal section of the kidney, within a very tiny area, these nephrons would be present. We will look at the size and shape, so there you will get more clarity in terms of how many nephrons can be fitted into that much of area. So these nephron consist of glomerulus and its associated tubules through which the glomerular filtrate passes before it emerges as urine. So this part we'll look at the when we talk of the function of or how the urine is being produced. But just to give you a brief idea, there is something called as afferent, or which is arriving. A stands for afferent, arriving. Arriving arteriole, that means they are bringing the blood to the nephron. And there is something called as efferent, which is called as or exiting. E stands for exiting. So that means that blood is going away from the nephron. So when there is afferent arteriole brings the blood to the nephron structure, and the efferent arteriole takes, these are the network of blood capillaries. What you can see is capillary network. That is what is glomerulus, right? Again, that might be hiding behind the let me just take it little down. Okay. So this particular structure is called as glomerulus. That is the network of capillaries which is arriving in connection with the exiting, that is afferent to efferent arterioles. That is called as the glomerulus. So this uh, network of capillaries glomerulus, and this C-shaped capsule-like structure is called as Bowman's capsule. Now again, it is shown in two dimension. Imagine a cup-like structure which is three-dimensional, which is like surrounding from all the sides of these blood vessels. So that is like the Bowman's capsule. So when so this point what it was speaking of the glomerular filtrate. So this nephron consists of glomerulus, that is the capillary network, and its associated tubule, that is afferent and efferent arterioles, through which the glomerular filtrate passes before it emerges as urine. So what happens now, I will explain a bit of mechanism as well here, because it will be important to understand this concept. So when the blood is flowing through this capillary network has a little pore size. So that means this network is porous. So the blood vessels here within the glomerulus, they are porous, because of which the blood will be filtered out. Now why it will be filtered out? There is a wide diameter here and there is a narrow diameter here. So because of which the pressure is built, and due to that pressure being built, the blood squeezes out. Blood as in the filtrate of the blood will squeeze out from this capillary network. It will be captured by this Bowman's capsule, and then it will flow down through the arrow direction. So that is what exactly happens in that first filtration system, that is through the glomerulus, and whatever filtrate is coming out of the glomerulus is called as glomerular filtrate, right? That is the first like step in the urine production, that we'll see in the mechanism for sure, but it was required to understand what exactly happens in the glomerulus. Now, they are microscopic structures. So these nephrons, they are microscopic structures which consist of a renal corpuscle and a renal tubule, right? Renal corpuscle means a renal capsule, which is this front part. So this glomerulus with the Bowman's capsule is called as renal corpuscle, because it is one of the capsular body or a bulb like body in the nephron structure. So it is called as renal corpuscle, and this other segment, which is a tubular, is called as renal tubule. So a nephron will consist of renal corpuscle, which is made up of glomerulus and Bowman's capsule, and the second part of nephron is renal tubule, which again have subsegments that we will see, specifically this proximal convoluted tubule, Henle's loop, this U-shaped body, and the distal convoluted tubule. So there are three segments of this as well. We will see them one by one later. As of now, I hope this part is clear. Nephron has two parts, renal corpuscle, renal tubule. Renal corpuscle is made up of glomerulus and Bowman's capsule, and renal tubule is made up of PCT, proximal convoluted tubule, Henle's loop, and DCT, that is distal convoluted tubule. The word nephron is derived from this word called Greek word called nephros, which means kidney, right? Which ultimately means a filtration system. And there are about millions of nephron in each human kidney. So if you look in terms of dimension, these are 35 to 55 millimeter long. 35 to 55 millimeter is 3.5 to 5.5 centimeter long, right? And they are squeezed in a very small kidney itself. Like kidneys are hardly 10 to 12 centimeter long, right? So within that, you can imagine how many of these would be fit at in the whole three-dimensional structure. So that is the length of the nephron structure. Now, at the one end, that is the at the renal corpuscle end, the tube is closed and is folded and expanded as well. So here the tube is closed, right? It is not open at all. You can see the Bowman's capsule end, this end of the C-shaped structure in blue, that is closed, and only through this network whatever filtrate is generated, that is absorbed into the Bowman's capsule. So it is expanded, it is closed, and it is folded also, folded in terms of C shape. And like I said, it is what you should imagine is three-dimensional cup shape and not just two-dimensional C shape. That is a representation. So, and it has a double wall, a cup like structure called as Bowman's capsule. Renal corpuscular, uh, or it is also called as renal corpuscular capsule, right? You might have to remember all of these names because they are just alternate names of different structures. So that is Bowman's capsule or renal corpuscular capsule. Now, this will enclose a cluster of microscopic blood vessel called as glomerulus. That is what we had seen. It is coming from the afferent arteriole, forming a glomerulus network of capillaries and becoming the efferent arteriole. And this we already seen. This capsule, that is the renal capsule or the Bowman's capsule, along with the glomerulus, is forming the renal corpuscle, which is one of the part of the nephron structure. Any question here so far? Anyone? I hope you are able to follow each of these terms, because later on you will have to remember in terms when we look at the mechanism there, we will have to remember what is where and how it functions. Okay, so that is all about the glomerulus or renal capsule along with the Bowman's capsule. So, oh, why this statement again? But anyway, I can skip the first statement. So, the glomerulus arises from the afferent arteriole. Again, these terminologies, they are very important. That's why they are highlighted. And then empties into the efferent arteriole. And like I mentioned, the blood will come from here. It will be squeezed out due to the difference in diameter. And because of that squeezed out, it will not be a blood as such. It is called as glomerular filtrate because it's kind of watery portion with the soluble components from the blood, and that will enter into the Bowman's capsule. So that is the function of this afferent and efferent artery. The smaller diameter of the exiting or the efferent arteriole helps to maintain the high blood pressure in the glomerulus. Now, why the blood pressure has to maintain high? So that the blood can squeeze out. It's a simpler mechanism. Like, when you have to water in your garden and sometimes you press the tip of the pipe, the water goes far away. That means the pressure has been built up, right? So that is the simpler mechanism that happens in terms of afferent to efferent arterioles. In terms of just the Bowman's capsule itself, it is divided into three different layers. So again, this capsule, which is receiving the glomerular filtrate from the glomerulus, this capsule is divided into three different layers. What we know as outer parietal layer, which is made up of the epithelial cells and with minute pores of diameter 12 nanometer. So they have to be a little porous so that they can receive the glomerular filtrate. So that is the outer parietal layer. Middle basement membrane, this layer is selectively permeable. Selectively permeable, again, that means it will allow the only few molecules to pass through it. So that's what is selectively permeable. This part we'll see in the function because there we will talk of different ions and so on. So there these things will come again. And the third layer in the Bowman's capsule is the inner visceral layer. This consists of large nucleated cells which are called as podocytes and which have finger-like projections called the podocell. Uh, Again, these are just to filter out something that is coming to the glomerulus, or sorry, that is coming to the Bowman's capsule. So outer parietal layer, middle basement layer, and the inner visceral layer. These are the three layers within this Bowman's capsule. The outer one, basement one, and the inner one. And this inner one will have the podocytes and the podocell, which are finger-like projection, which is nothing but the like outgrowth, hair-like outgrowth of this podocyte cell. Again, any question, anyone?
Yeah. So the afferent and the efferent artery have blood in that.
Yeah.
The blood is filtered out.
Blood is filtered out.
So then where does the water come in?
So from the blood.
No.
So if you separate the RBCs from the blood, what you get is plasma. RBC, WBC, all those cells, right? So whatever filtrate is generated in here, that is actually your plasma filtrate. Cells cannot pass through this blood capillary network.
So basically the plasma comes out.
Yeah, but then that's when I call it like there is specific term glomerular filtrate, which is just like you know, if you have milk which is kind of spoiled, right? What ultimately gets out is water with some soluble components of the protein, something like this. But this is not spoiled for sure.
But the blood it might get thicker.
It's not. It's how can I give you that analogy? It's like you have a pipe structure, and let's say you have some filter or mesh like network on that pipe at some places. So some content will squeeze out and the rest will go back, right? It will continue to flow, and then at few other points it will again reabsorb the water to some extent. And plus that's when the maintaining of the balance of the fluid as well as salt is done by the kidney. How much water should come in, how much water should go out of the body, that is decided by this nephron structure. So blood doesn't thicken as such. Okay, so that was the renal corpuscle. Now coming to the renal tubule part. So in case of renal tubule is a long convoluted structure. Convoluted means folded, right? If you look at this diagram, it has many folds like this kind of wavy structure. That is what it means by convolution. Convolution literally means folding, right? Now again in two dimension, what you are showing is just like up and down, like a wave like structure. But in three dimension, it can be any type of convolution. It can be folded in like kind of a circular manner and so on. So that way these PCT and DCT they are present. Let me just check if I can show you the nephron structure original. Nephron real image. Again, I assume, right? No. Yeah, this is something kind of it, but not literally true. So this image, I can't figure out where is the glomerulus. Yeah, probably somewhere here, this blue thing also. But again, I'm not too sure because the diagram is too small. This is the actual kidney image, but again, looks too creepy. Yeah, this is actual perfect image. Now, if you look at this particular part, you can see this is the glomerulus, right? This is the cortex, right? This is the cortex. This is the medulla part, and the glomerulus is shown here. So there will be one of these will be the efferent, most likely this one, because you can see it is branching from here and it is going into this nephron, and the exiting one is going here, right? So that way this is glomerulus. This part you can see the convolutions or the folding. It is not at all like what you see in the diagram. So the convolution means folding, and it can be any type of folding. And then there is something called as Henle's loop, which we will see. Okay, let's first look at the structure, and then we'll come back to that diagram. So, so the renal tubule is a long and convoluted structure that emerges from the glomerulus, right? Obviously, it is a continuation of the glomerulus or the Bowman's capsule, and can be divided into three parts, and this is based on their function and of course on the structural aspect also. That is the first part, is called as proximal convoluted tubule. Proximal means front or forward. So that is the first part of the folded tubule, that is proximal convoluted tubule, short form as PCT. Due to its proximity or due to its closeness to the glomerulus, that is why it is called as proximal convoluted tubule, and it stays in the renal cortex, that is the outer part of the kidney, or the outer layers of the kidney. That's where the PCT is present. The second part is called as loop of Henle, or it is also called as nephritic loop. This was identified by this guy called Henle, and that's why it is called as Henle's loop. And because it forms a loop, that is a U-shaped structure with descending and ascending, that is going down or going deep into the kidneys and coming up towards the cortex, that is ascendingly, and that goes through the renal medulla. So this I want to show you here. Again, I don't know if I can zoom out this. Let me show you here. Yeah, if you look closely, so this is the PCT, whole of this broad thing up to here would be the PCT, proximal convoluted tubule, starting from glomerulus, then it ends up into the Henle's loop. This is the descending limb, the first part, and then it goes ascending, that is upward, and then it ends up again into further coil. So that is what is the Henle's loop. And if you see the PCT and glomerulus, these are all within the cortex part, and this one Henle's loop is within the medulla part. So that is how they are differentiated as well. And there are of course certain functions of being present there that we'll see in the mechanism part. So the second part is the loop of Henle or nephritic loop, and because it forms a loop with the ascending and descending layer, that goes through the renal medulla. Now the third part, which is the renal tubule, is called distal convoluted tubule. Distal, again a little far away or farther from the glomerulus, and that is why it is called as distal. Convoluted because again it is folded, so distal convoluted tubule or DCT. And this part is restricted again to the renal cortex. So the nephron starts with the renal cortex, the Henle's loop goes into the renal medulla, and it again comes back to the renal cortex with the DCT. So that is from outer layer, the first two part, glomerulus and PCT, is in the renal cortex. And the third part, that is the Henle's loop. So glomerulus, PCT is in cortex, Henle's loop in medulla, and again DCT, that is in the cortex region again. So that is how this nephron is arranged within the kidney. Now the capillaries of the glomerulus, they are enclosed. I don't know why this is coming again. Sorry, my bad. So that is the Bowman's capsule that we know. So yeah, and also it's like in between nowhere. I missed it probably. So the structure, that is this structure as in the DCT, extends to form highly coiled tubules. Sorry, people, something is wrong here. I must change it right away. Uh, it is not this. Okay, it's in continuation to this. Okay, then it is perfect. So it's talking of the Bowman's capsule, and this Bowman's capsule extends to form PCT. PCT continues to form loop of Henle, which ascends to DCT, and this will open into the collecting. Now it is correct. Yeah, yeah, because I gave wrong punctuation there. So the collecting duct is actually, the name suggests, it is kind of main branch of the all the nephrons. So again, if we go back to this part of the nephron structure, this big tube-like structure, you can see, and it has this entry points, right? These ones are the entry points for the end of the nephron, that is the distal convoluted tubule will enter into the collecting duct, and that will collect all the urine or all the filtrate that is formed, and then it will take it to the renal pelvis area. So that is the main structure of this nephron. Any question, anyone? Okay, so now going little bit towards the functional aspect of all these three structure. Uh, the major function of this tubule is the reabsorption. Reabsorption in the sense whatever is gone as a filtrate from the blood has to be reabsorbed, because a body cannot uh, like take that loss of those many nutrients in one filtrate, right? So that is when they need to be reabsorbed, and the process can either be through active transport, that is with the help of energy, or passive transport, that is by the process of diffusion also. So in addition, the secretions by tubule help in the urine formation without affecting the electrolyte balance of the body. So tubule has two actions. One is reabsorption and the other one is secretion. Secretion means, like let's say there is excess of particular iron or particular salt within the kidney or within the blood, that will be filtered out for sure. So from the tubule itself, it will be again filtered out, or it will be again reabsorbed depending on whether it's more or less, and then it will go into the urine. So if it is more, obviously it will not be reabsorbed, rather it will be secreted and it will go into urine. If it is less, it will be reabsorbed and it will not be secreted. So depending on what is the concentration, this electrolyte balance of the body will be altered based on the nephron function, either as a reabsorption or as secretion. Now, in the proximal convoluted tubule, the blood is brought by the renal artery, is filtered by the glomerulus, and passed on to PCT. This part we have seen, and this happens due to the filtration pressure, or the pressure between, or the difference in the diameter of the afferent and efferent, and that is why the blood is kind of squeezed out. That is why the pressure is being built up and the filtrate is generated. That filtrate comes into PCT, proximal convoluted tubule. Maximum reabsorption takes place in PCT. Maximum, because that is the first lot of the filtrate, right? From that, whatever is required, especially glucose, sodium chloride, or most of these proteins, amino acid, they are reabsorbed in the PCT. So it includes like glucose, proteins, amino acid, a major portion of electrolytes, and water takes place. So that is when your question, water also gets reabsorbed in the PCT itself, right? So the surface area for reabsorption is facilitated by the lining of the simple cuboidal epithelium. So within the nephron structure, there is this cuboidal epithelium which has a large surface area for increasing the absorption or reabsorption process, and reabsorption takes place at the expense of energy, that is it is an active process, because it will take a lot of energy to reabsorb all the molecule again. Why? Because it will be against the concentration gradient, that is from low concentration to high concentration. Blood at any given time point will always have very high concentration, right? And because of that, these molecules, they will have to get reabsorbed into the blood, and that has to be the active process. So that is the reabsorption in the PCT. uh, so this proximal convoluted tubule, it selectively secretes ions such as hydrogen, ammonia, potassium into the filtrate. Again, I hope you are able to understand these two different processes. One was reabsorption of many substances like glucose, protein, amino acid, electrolytes, and water. And the second function that PCT does is the function of secretion, that is excess of hydrogen, ammonia, potassium, and it will absorb the bicarbonate from it. So that way these different ions, they are going into the filtrate, and the bicarbonate is taken back into the blood system, and thus PCT maintains the electrolyte and acid base balance of the body fluid, being one of the major organ or major part of the nephron, which uh, reabsorbs as well as secretes at the same time. That's when it is able to maintain this electrolyte or acid base balance. Acid base balance because it maintains the concentration of H plus ion as well as NH3 or NH2, uh, like NH4 plus ions. So that way, and also the bicarbonate that way. So that way it will maintain the electrolyte as well as the acid base balance. Any questions? Anyone in the first part?
Does the absorption and secretion happen at once?
Not really. It happens at selective segments. So it's always an exchange process. Like whatever is needed, uh, it will be taken back and it will be exchanged with whatever is not
It's the same thing.
through the same portal. But that is not, we can't really call it like it happens at the same, it happens at the same time, but it's not the same process. You get it, right?
Yeah, like you give and then you
Yeah,
get.
correct. Okay, so that is the function of PCT, that is proximal convoluted tubule. The second part, and I guess we will stop after this, is the Henle's loop. Now, with the Henle's loop, there are two limbs, like I showed you in the diagram. There is descending limb. Again, most of the students, they get confused which one is descending and which one is ascending. Obviously, even if we look at this diagram here, so from glomerulus to Bowman's capsule to PCT, it is going down and it has to be descending. So the first uh, limb of Henle's loop that comes is the descending limb, and then there is a U shape, and then there is ascending limb. So the descending and the ascending limb of the Henle's loop, uh, being part of the same loop, both ascending and descending limb show different permeability. Different permeability means they both of them they show difference in terms of what they absorb and what they let go of. So the descending limb, that is the first part of the Henle's loop, is permeable to water but is impermeable to electrolyte. That means water can squeeze out through it, but the electrolytes cannot. While the ascending limb is permeable to electrolyte but is impermeable to water. So again, let me go back to the diagram. So this first part will allow water to go out because that's what forms the major content of the urine, right? So the first part will allow maximum of the water. Another thing that we must remember is the diameter of this tube is shown here common, right? Throughout this renal tubule, it is shown like same. It is not same. It is exactly like this. You can see this has kind of less diameter. This PCT has bigger diameter. Henle's loop has thinner diameter to some extent, and they again go for higher diameter, and then it continues like that. So that is how these structures are present. But like I said, this is the representative diagram. So that's why they might have shown the same diameter throughout the renal tube. So what we are talking of the Henle's loop. So the descending limb, that is the first part, is impermeable to salts or electrolytes. So from the first part, because the salts and what we call as these electrolytes, they will be required. So they will be reabsorbed later into this capillary network. You can see these blood vessels around the Henle's loop. They are absorbed in there later. So first part is permeable to water, and so only water can go out from the descending limb, electrolytes cannot. And the second part, that is the ascending limb, it will allow the water, so it will allow the electrolytes to pass through and not allow the water to pass through. So that is impermeable to water, permeable to water, impermeable to salt, that is the descending limb, and it is permeable to impermeable to the ascending limb. So that way this statement speaks here: the descending limb is permeable to water but impermeable to electrolyte, while the ascending limb is permeable to electrolytes and impermeable to water. So at one place only water will be allowed to go out, at one place only electrolytes will be allowed to go out. So that way this Henle's loop function. Now since the electrolytes gets reabsorbed at the ascending loop, right, because it is impermeable to electrolyte, that means to allow, not allow them to go out, but they can get reabsorbed through the mechanism of active transport. So since the electrolytes get reabsorbed at the ascending loop of Henle, the filtrate gets diluted, right, and as it moves towards the ascending limb, but reabsorption is limited in this segment. So what it is meaning is, because it is, the descending limb is impermeable to water, so water stays as it is, and water goes out of that first part and it becomes concentrated because it is impermeable to electrolyte. So less water, more electrolyte, while in the other situation, that is the ascending limb, it is permeable to electrolytes. So that means electrolytes can go out and impermeable to water. So the amount of water stays and the amount of electrolyte is reduced. And that is when the filtrate gets diluted as it moves from descending to ascending limb, right? And here the reabsorption is limited in this segment, that is the ascending limb. In descending limb, that is the first part, reabsorption can happen of the electrolyte, but in the descending limb, so in short, whenever maximum reabsorption happens here, second maximum or second lower grade of reabsorption happens here. As we go further towards this collecting duct, the reabsorption goes on reducing and the secretion goes on increasing, right? So that is the general mechanism. So for today we will stop here. Next class we will continue with the distal convoluted tubule and the collecting duct. For the time being, you can go through uh, these slides in terms of function, or I will share a video if I find one, uh, which will talk about this function. But these things will again get revised when we talk of the mechanism of the urine formation. So there we have to talk of all these things again. Here it is structural requirement in terms of permeability and so on. So that's why we need to discuss it here. Okay. Any question? Anyone? So much of silence.